not one single state. They are complex states. They kind of keep changing between each other. Once you start giving one drug, the real relative accessibility of each molecules actually change. And therefore, one should stop looking at diseases as an analyte space. It should be looked at as a state space. And what that means now is that rather than looking at which biomarker is abnormal in an analyte space, for example, you try and study biomarkers, either it's an mRNA biomarkers or any biomarkers that you measure, and you're able to measure and you try and correct that and say, oh, this is the right thing in the disease perspective. From a state space, the whole perspective is completely different, and it's really got to do with which biological state is a patient occupying and how is it evolving actually. And I think that's the real perspective of what my lab wanted to do. And about 4 years back, in fact, when I started shutting down my lab, that's what our entire focus was. And I will talk about how I believe liver has a very, very important and strategic role to play from the context of how liver manages multiple states and diseases actually. But I think this is a concept that I hope you're able to appreciate. For example, if you have two different people with sugar levels, if you measure their sugar levels, and both of them have normal sugar levels, but it's not necessary that both of them have an equally normal biological state. The biological state may be different and the perturbations must be very different. And therefore, the way we look at diseases today is from an analyte point of view. What we really need to look at from a state-specific point of view, and how we'll be able to do it. And in fact, the video that I shown earlier, which talked about building digital twin models and using AI-based models, is the only way I think I can perceive that future goals and models can be identified and set up there. And therefore, Department of Biotechnology is also planning to set up large number of digital twin centers across the country, where the health measurements can not be single parameters, but complex body parameters of system levels understanding, what we really try to do. From the point of view of liver, let me just come back to the key perspective where what I wanted to give. I mean, the gut-liver axis is really well understood and one of the key perspectives that we constantly worry about actually. And I'm sure a large number of ILBS faculties and students really look at the gut-liver. However, this bidirectional crosstalk that is present there through the biliary tract, portal vein, and systemic circulation actually, how does it controls and what are the kind of aberrations in the gut that leads to an altered homeostasis in liver is a question that my lab started asking about 5-7 years back actually. Is that if there is a perturbation in the intestinal gut, does this communicate to the liver? And if you communicate to the liver, how does liver respond, liver cells respond? And therefore, can you identify therapeutic targets by which you can correct these diseases that are intestinal diseases? And one of the most common, these studies were initiated by two students, actually, Jyotsna and Binayak Sarkar. Some of you may even know who've been there for a while, actually. In fact, we published this paper, and I'm not going to go into details of it, but actually, if somebody wants to take a look at this paper, I actually think it's an exciting way to look at how hepatocyte-specific transcriptional program, which is mediated by two key transcriptional factors in the hepatocytes, controls experimental colitis in mice by modulating bile synthesis. So it talks about disease models, it talks about enzyme targets that modulates it and the mechanism by which it does it actually right now. And I'll just show you what the working model of this is actually. So typically, FXR signaling in hepatocytes control the CYPs, and these CYPs then make primary bile acids, and these bile acids are secreted to intestine and other cells, like various other parts of the body, and where they maintain a homeostasis. If you have an inflammatory state, for example, typically you can colitis model, if you try and use a colitis model of intestinal inflammation, the trigger happens somewhere else. But the reset in the organs are at multiple levels. And while we keep treating the inflammation in the gut, actually, we forget that there are other things that have happened. And in this model, what we establish now using, of course, it's in the mice state. We got back some data in humans as well with collaboration with Vineet, actually, from All India Institute of Medical Sciences. But what we showed here, that in fact, the CYPs, which produce the bile acids now, get activated and produced through a completely new pathway, which is through Relay and STAT3. These are the two important transcription factors which are completely silent in the normal conditions. So if you take a hepatocyte specific knockout of Relay and STAT3, the mice has absolutely no issues. The mice is perfectly happy. But only under inflammatory conditions that both these markers actually gets activated, and if they are not present, it actually impacts the gut status. So what we showed here, that the primary bile acid gets accumulated through a completely different pathway, and this pathway then leads to enhanced damage epithelium cells and heightened immune response. And so this entire cascade of events gets produced by a inflammation that is caused in an intestine, an altered transcriptional response in the hepatocytes, which resets the pathway such that the bidirectional crosstalk allows it to work in the system. And through increased bile acid synthesis, it brings back the inflammatory state, and therefore it maintains what we call feed forward loop. And I'll show you this feed forward loops are the very, very important perspective of human diseases, actually. And I'll show you this little bit in another model. And if you really, as I said, if you want to see, you can look at this paper that's published in this in eLife. So how does the human liver is as Dr. Sarin always used to tell me, actually constantly, ever since for years, I have known him now for almost 20 years now, that liver is the brain of the body, actually. All another brain or some word you used to use, I remember, it's the central brain of the human body or something like that. And I think the way reason it is it controls the metabolic hub of the entire human body perspective, actually. And I think that's the model that it's involved in synthesis, secretion, degradation, and regulation, which are the key biological processes that control our human body system, entire system of the body. And every aspect of this is controlled. So even though you think pancreas is the one that is important for maintaining the blood sugar level, the liver state is equally important. The gluconeogenesis that happens in the liver state and the activities that happen that we hardly worry about, actually. Similarly, the secretions of hepatokines, which is so important in controlling variety of factors that are controlling various organs, degradation of fatty acids, and the regulation of metal ions, that is so critical and important. So how do we start studying these kind of models? And I had interest in, as I said, a model to study is an area where you don't have much competition, actually. And that time, I realized that one of the area that was really interesting and exciting to me was what is called cachexia, actually. And some of you may know this. Cachexia is basically a multifactorial syndrome. That means I don't understand much about it. And therefore, what it looks at is basically loss of skeletal muscle mass without the loss of fat mass that cannot be fully reversed by nutritional support. So you try and treat a patient and cure him from the disease, but in many cases you actually get cachectic response, and this cachectic response is a response of the body where you have not been able to set its metabolic functions right, actually. And these abnormal, so even though the body is variable, a combination of reduced food intake and abnormal metabolism, actually. So it's really systemic catabolic syndrome and profound consequences for patient prognosis. For example, wasting, chronic illness, infection, malignancies, and many transplants have this problem, actually. Or if you look at the cancer chemotherapies, you do give chemotherapies, and post chemotherapy, you are not able to improve the patient's health because of the consequences. So there are two kinds of cancer-mediated cachexia, which has now become very common and popular to really look at. The second one that I thought that I should study about is an infection-associated cachexia, actually. And some of you might have, had the thought process of coming during the COVID times when you would have an infection, but you will feel so weak that you will not be able to, just work around, and it took years for you to recover from it, actually. And so this is the cancer cachexia. People thinking about how complex networks are set up. There's a malignant inflammation, tumorokines, metabolic mayhem, interorgan crosstalk, and systemic dyshomeostasis is what causes cachexia, actually. And that's where the whole scenario of medical intervention, therefore, as I said, requires a digital twin models to look at. The one that I got interested in because of my interest in TB was consumption began with a cough, actually. And this is a classical way the TB used to be treated in 19th century. Tuberculosis had another name called consumption, actually. It was never a TB, actually. People did not know what causes TB and all. And in fact, TNF alpha, which is a primary cytokine, was known as a cachectin factor. And clearly, the TB is clearly associated with fatigue, excessive weight loss, anorexia, and general wasting away of victim. And I'll show you. And what I wanted to ask the question is that because of this really central role of liver in all these aspects, is there a TB? Can the TB have a liver specific symptoms actually? So if you really look at classical symptom of TB is like cough, which is a coughness. However, if you really go back and deep into the TB pathophysiological changes, you have bone degradation, you have muscle wasting, you have weight loss, and you have micronutrient balance. And what broadly it tells you that there is a breakdown of immunometabolic network, and therefore the central hub must be a role to play actually in this disease perspective. So I started asking a simple question in the lab, and I asked, does the mice infection model of TB in fact has a liver infection anytime it infects the liver and therefore perturbs the response or not? That's a direct way of looking at it right now. And so you do these experiments, you aerosol infect mice and follow them with this analysis. And what you will see, while the lung infection begins from the day one, in every single mice, in the 4th week, you have liver infected with TB infection, actually, Mycobacterium tuberculosis infection. And this was reproducible across every single mice. And this was not an instance of miliary TB, what many of you might have read about, where you say the TB is spread to all the organs and therefore it is happening everywhere. It's not that case. It only goes to spleen, and from spleen it migrates probably to liver, and we still don't understand how does it work. But we wanted to understand the whole concept of TB cachexia, because why does patient lose weight? Why does patient get anorexia? You have an infection in lung. You should be able to take care of that part of the body actually. But the real challenge is to understand this cross-talk connections and how to be able to take this up. And I think in the end, I'll try to explain you that this is not sufficient to understand the TB only from a TB point of view, why you really need to. So this is a histopathological changes in the liver of the mice post-MTB infection, and you can very clearly see the infected mice livers has lots of inflammation and various structures that are induced in the mice. And clearly the classical pathophysiology of hepatocytes is actually get altered. The question was that one of the key questions that you will ask for Kupffer cells are the macrophages in liver, and therefore the liver infection may be due to the macrophage cells because TB is known to associate with the liver cells actually. And therefore we try to understand whether it is really the hepatocytes that where the infection is there, or is the other cells that are these endothelial cells, lymphocytes, Kupffer cells, and stellate cells are the infected involved in it. So what we did was I infected mice. We isolated the pure hepatocyte population, and you can characterize them. And you can see nice cells under the microscope, as well as you can characterize them using FACS staining as well. And we clearly showed that Mycobacterium tuberculosis is in fact present in these cells only, hepatocytes. And the numbers are fairly large. So what that means is that Mycobacterium tuberculosis, for reasons that is unclear and not established till today, even in a classical mice model where every discovery program and all drug discovery programs happens actually, and in the 4th week itself, 100% of the time you find in the liver cells of TB cell Mycobacterium tuberculosis infection. So the question was, can you find this in human biopsy samples and study them? In fact, you can see that these are various biopsies of humans we collected from PGI Chandigarh, and these were obviously post-mortem cases where we collected these samples with the help of Dr. Ashwini Kumar and did access FACS staining and demonstrated unambiguously that many of the TB patients who have died of tuberculosis, in fact, have quite a bit of infection in their liver perspective from a liver point of view. The question then is what is that is going on in hepatocytes and MTB infection, and why we never care about it. I mean, TB being such a prevalent disease and so many people working on it, why have not we ever looked at it actually? And it is very interesting to see that the kinetics of MTB infection and hepatocytes, the lower panel shows what happens in a macrophages. If you infect macrophages with TB in an ex vivo model, it actually grows first rapidly and then slowly starts to kind of stop the progression of the disease because it forms granulomas, and macrophages actually contain the disease in a very small period of time. if you really look at in hepatocytes, this is the hepatocyte data, and I don't need to even quantitate the data. You can see how rapidly the Mycobacterium tuberculosis, which is labeled in green, is in fact growing. And in fact, the numbers after 120 HPI, 120 hours after HPI, the hepatocytes allows Mycobacterium tuberculosis to really grow very, very rapidly. So it's in fact a favorable niche, unlike macrophages, where macrophages is able to blunt the response of Mycobacterium tuberculosis. The hepatocytes are in fact a favorable niche for TB to survive, with both in primary hepatocytes as well as HG2 cell lines and a lot of other cell lines that we tested out actually. So you can see the data is quite remarkable actually, why the Mycobacterium tuberculosis in a classical macrophage model, which is BMDMs or THP1, actually go down with time. In fact, in hepatocytes, they continue to multiply and continue to grow faster. So, I won't go into the details of how it adapts and all, but many of you would realize and remember, if macrophage biology, if you have studied, that one of the key perspective of tuberculosis is that it uses lipids as its carbon source when it's in an infection stage. Rather than using glucose as a carbon source, it uses lipids as a carbon source. And macrophage is a lipid droplets that is used as a food for growth. And that's the reason why hepatocytes, all of you will understand this, the hepatocytes have the largest number of granules, actually, lipid granules. And that's why, many of diseases that happens actually. And in fact, the data to show that that lipid granules really, it eats up these lipid granules and survive very, very well, in fact, in the hepatocyte cells actually, and therefore TB is able to survive and do well. So the question that I wanted to ask then after this was, what happens when Mycobacterium tuberculosis resides in the hepatocytes? Because as we discussed about these SIPs, which are very critical and important, one of the key factor in one of the key SIP in one of the treatment of Mycobacterium tuberculosis is a classical drug called isoniazid, which is a pro drug, and it gets activated in the liver, and then that activated drug, pro drug, conjugates with NADP, and that's the active form of a drug actually. So many diseases, in fact, get activated as the drugs go into your body, and that this pro drug works. So the question to ask was then is that if I have hepatocyte cells infected with Mycobacterium tuberculosis, do they impact the TB drugs susceptibility to rifampicin and isoniazid? And rifampicin actually does not require activation of liver, and isoniazid requires an activation of liver actually. And this is a very important problem to understand, because if you really look at our TB load in our country, most of the TB patients that reaches tertiary care hospitals have already gone through a disease pathogenesis for almost 5-6 months of various treatment that they take, and they carry out activities. And in fact, even if you say 4 weeks is a long enough time in mice models to bring, you can almost imagine that 3-4 months is a good enough time for the Mycobacterium tuberculosis to escape and reach the liver infection model. And therefore, if you really look at data as compared to macrophages, hepatocytes provide a drug tolerant niche to MTB. And in fact, we were able to show very beautifully in this data actually that what happens is that the anti-TB drugs after 24 hours were given, while rifampicin has no impact on it, the isoniazid clearly had an altered response actually. Now, drug metabolizing enzymes are upregulated both in vitro and in vivo infection conditions. So one of the key perspectives then is to talk about the DME enzyme that is present in hepatocytes. So we demonstrated both using an in vitro and in vivo model that these DMEs are upregulated by MTB infection in hepatocytes and liver. And therefore, whatever dosage of isoniazid that we provide to patients who come in tertiary care hospitals, in fact, is likely to be suboptimal. And in fact, what this will drive is a kind of a program that will make more drug resistant bacteria to it. And in fact, if you really go and ask all the microbiologists in the country, you'll see more than 40% of the drug resistant strains in TB in the country belongs to actually isoniazid resistance actually. And I think that's where probably one of the impact. And you can do these assays and you can demonstrate that there is a IC50 is clearly shifted, and this has got nothing to do with the drug tolerance of MTB inside hepatocyte, but is it independent of an intrinsic factor? Is because of the hepatocyte's growth, it is actually doing this right now. And then we wanted to show that this enzyme activity is really activated in vivo as well. So we isolated enzymes from the mice, and set up a mass spec based assay to demonstrate that the NAT2 enzyme, which is the one that enzyme that converts isoniazid to acetyl isoniazid, which then gets activated further in the presence of, which is important for as a pharmacological agent, the higher production of acetyl isoniazid and MTB infected HepG2, you can see, is fairly clear. And therefore, what I'm trying to conclude from all these studies is that hepatotropism of Mycobacterium tuberculosis during the chronic infectious cycle results from an immunometabolic dysregulation, actually. And that's one of the classical models that we need to think about, which magnifies local and systemic pathogenicity and altering clinical presentations. And I think this is very important as I'm trying to talk about. It's not only chronic diseases have multifactorial manifestations. Even infectious diseases have multifactorial manifestations. And we've underplayed our own understanding of these diseases perspective. In fact, COVID was a very good changer for changing this paradigms because people who had chronic diseases, they were actually had more and more problems with COVID actually. And what hepatocyte does, MTB shields itself against the common MTTB drugs inducing drug metabolized enzymes, and therefore it survives in hepatocytes. It is not able to be getting killed with the isoniazid that is present there. And then the replication conducive milieu of lipid droplets in hepatocytes actually upregulate through a PPR gamma, which I did show you data actually, which is a transcription factor that regulates all of this and brings down to the host cells actually. So I think just to conclude my talk and give you a perspective that liver has a really, really important role to play in a lot of disease perspective actually. And this immunometabolic organ, in fact, is a central metabolic hub, an immunological hub that controls large number of factors. One I showed you in a classical example of gut-liver axis. The second was an infectious disease where we've gone and identified the mechanisms that it modifies. Because just showing that the presence of bacteria is not sufficient. And so what happens is that it infects lung, then through lung it is transmitted to liver, and as of now we have not had time to really figure out how does it do it. In hepatocytes, it forms early endosomes, and these endosomes actually produces lipid droplets. These lipid droplets, it starts feeding on these lipid droplets, larger lipid droplets actually comes about, which changes in effector functions of metabolic pathways, secretory proteins, and hepatokine secretion. And one of the reasons the hepatokine secretion is one of the classical hepatokine secretion is the one that controls the weight of the human body actually, which is GLP-15 actually. Many of you would have looked at these hepatokines. Now this alters the entire homeostasis status of human body. And therefore you get cachexia, you get metabolic disorders, and you get drug inactivity. And I personally think that these are the kind of complex interorgan crosstalks that we all need to understand if we need to start curating diseases and start solving problems rather than just staying with, managing the diseases. Because there is a bug and you need to remove the bug, and therefore you only give an antibiotic treatment to a patient, is an insufficient way to solving problems. It solves the problem, but it does not curate the problem actually. And I think that's where all of us need to bring our best together. And I think, as Professor Saran showed, how various IIT professors and students are interacting with you to develop these digital twin models. And I think the future of medicine is through the integration of IT-enabled digital twin models, which can almost say that, can I have a replica of mine? Can I model myself? Can I use various parameters and use them to optimize and therefore provide a clinical advice, which the clinician then can use and can actually take it further ahead. And I'm sure the Department of Biotechnology will be more than excited to work in this area of chapter and work together in teams to be able to do this. In fact, we are going to announce a call to set up a center of digital twin in another couple of months to be able to across various diseases. And it's not just diseases only, but digital twin models require for sustainability models, for agriculture, for various aspects where complex problems require solutions that can be addressed by human things. I'll just end by my lab students who do all the work and I do all the talking. And I think it's such a great pleasure to have such fantastic students who work on these complex problems that they identify. And thank you very much for all of your time and on Saturday morning coming here to listening to me. Thank you very much.
It was an outstanding talk, and I learned and lots of notes, and I realized that you can be a bureaucrat, but you can remain a core scientist. A big round of applause for Dr. Berkeley. And we are very privileged that you have the experience and vision for working on hepatocytes and also that stimulates us. For digital twin, one simple example could be a patient has a liver tumor, another patient has a liver tumor. This guy develops faster